Dear students, in this part of video, I will discuss about auxin and its mechanism of action. A classic response to auxin is cell elongation. And cell elongation is induced because of acid-induced cell wall loosening. The model which best describing this process is known as acid growth hypothesis. As we know that auxin is a growth hormone and the growth of the cell totally depends on auxin. The growth of plant cell depend on auxin. Auxin activates proton pumps which are present in plasma membrane. In previous video, I have discussed how protons are formed. Protons comes out into the cell wall. Proton come into the cell wall, which makes it acidic, and the pH level maintained is 5 to 5.5. Because of the lower pH, some specific type of growth enzymes which are known as expansins becomes activate. These enzymes hydrolyzes the hydrogen bonds within cellulose. As we know that cell wall is made up of a polysaccharide known as cellulose. And hydrogen bonds between these microfibrils are broken by these expansins. After breaking these bonds, loosening of cell wall takes place and cell can slide easily, due to which the water uptake takes place. As water enters into the cell, the passive increase in the cell size takes place due to the increase in the internal turgor pressure of the cell. Auxin intact at gene level by synthesizing new proteins or enzymes. These enzymes are required for synthesis of new cell wall material. Cell elongation in response to auxin. We can understand it by this slide. Auxin increases activity of proton pump, and due to the increasing concentration of protons in the cell wall, it becomes acidic, due to which some specific type of enzymes which are known as expansins, due to which the specific type of enzymes which are known as expansins become activate. Expansins are responsible for cleavage in cellulose microfibrils from polysaccharides, and exposed polysaccharides now accessible to enzymes. Enzymatic cleavage of polysaccharides allow microfibrils to slide. Enzymatic cleavage of polysaccharides allow microfibrils to slide. Cell wall can extend due to the increased turgor pressure. The outcome of acid growth hypothesis is cell wall loosening is mediated through a specific type of proteins which are known as expansins. Once the cell wall is loosened, water moves into the cell, which is driven by higher internal osmotic pressure, which causes the cell to expand. Auxin interact at gene level by synthesizing new proteins or enzymes. And these enzymes are required for synthesis of new wall material. By this graph, we can understand how indole acetic acid or auxin stimulates proton pump. By this graph, due to which expansions become activate and cell elongation takes place, which ultimately result in the growth of the plant. This is how auxin play its role in cell elongation or growth of plant. Thank you very much. If you like my video, please share, comment and subscribe it.